So my name's Rhian Convery. I'm a postdoctoral research fellow at University College London. And today I'm going to talk to you a bit about the GEMFI study, which is our genetic study of FTD, and some of the other digital projects that we're now starting that also looks more deeply into testing people with FTD and ALS. So just a bit of a brief background of what I'm going to cover. So I'll start just by talking a little bit more about FTD and how this condition overlaps with ALS, as we all know, but also the work we do as part of GEMFI before moving on to some of our EDOF digital studies that were conducted in both FTD and ALS. And I'll just finish up by discussing some of our more exciting future projects that we're going to start hopefully very soon. But first, just to give you some context to where I work. So this is Queen Square in London, which is home to the National Hospital of Neurology and Neurosurgery, or the NHNN. And it's the UK's largest dedicated neurological and neurosurgical hospital. And within this hospital, there's a cognitive disorders clinic where we have about 10 cognitive neurologists that work and see patients with different conditions, but primarily there's a particular interest in young onset, atypical and genetic forms of dementia. And what you can't see is that this is the Dementia Research Centre, but what you perhaps can't see from these two images is that these are across the square from each other. So these two places actually face each other in Queen Square. And this is a department within UCL, which is where I work. And we have over 30 years of patient-focused research into different dementias, and we have a number of observational cohort studies, including GEMFI, which investigates familial or genetic FTD. So that's Queen Square. But just to give you a bit of a background to FTD, so under the age of 65, FTD is actually the most common young onset dementia. It also occurs in those over the age of 65, but Alzheimer's disease and vascular dementia are much more common in this population. The age of onset of symptoms is typically in the 50s and 60s, though in some cases it can occur much earlier, as early as the 30s, or even much later in the 70s. And FTD is actually an umbrella term that describes different conditions that can affect your behaviour, which is called BVFTD, but also your language, which are termed the primary progressive aphasias. And in around a third of people, symptoms of FTD are caused by a genetic mutation that is autosomal dominantly inherited. And this means that each individual has a 50% chance of inheriting a gene from a parent that is also affected. And there are many genes that have been shown to actually cause FTD, but the three main ones that we talk about the most often are MAPT or tau, C9orf72, and progranulin or GRN. And I'm sure we all know here today, there's been spoken about a lot, that C9orf72 is also the genetic cause, or one of the genetic causes of ALS. So this is how it overlaps with FTD in this condition. So that is a brief introduction to FTD, which leads us on to GEMFI. So GEMFI stands for the Genetic FTD Initiative. It's a European and Canadian multi-centre cohort study with the mission to follow families with genetic FTD through their life in order to detect the earliest changes that occur. So, with the main aim really to sort of better inform treatment trials and think about the best measures. And GEMFI's been doing this for a while now. So the study was set up in 2012 with just a few research centres across the UK and also in Rotterdam. But there are now 47 contributing centres across 14 different countries in Europe and Canada. And within the study, we've almost seen 2,000 participants. I wish I could have come here today and said that we'd reached 2,000, but just not quite. Over 5,500 research visits over the past 14 years. And as Emma very nicely introduced in the previous session, we do a lot of tests and we collect lots of different data across a wide range of measures when someone comes in to see us for a research study. So for example, we collect a detailed clinical assessment alongside neuropsychological testing. We do an MRI scan, collect fluid biomarkers, and also collect genetic information across our cohort. And much like ACORN, we do this in an anonymised way so that people don't need to know whether they carry a gene in order to take part in our study, as long as they have a first-degree relative that is affected. But more recently, the study has really started to incorporate digital technologies and wearable tools into our research to think about how we can collect more frequent data from people from the comfort of their own homes. But I'll talk a little bit more about this later. So, as I just touched on briefly, GEMFI is a study of genetic FTD families. So we see individuals who have symptoms of FTD and have a diagnosis. But we also see those individuals that don't have symptoms but are at 50% risk of carrying the gene. So they have a first-degree relative that is affected, which means we have an entire group of non-affected family members, some of which carry the gene and some of which will not. And we see mostly individuals from families that carry one of these three main genes that I talked about, but there are other rarer genes as well that cause FTD too. And as we have a number of people in the study with C9orf72, we now also see people that have ALS as part of our study. And we've been collecting this data from individuals over the past 14 years. And as I said, we've got almost 2,000 people, and almost half of those individuals are participants from C9orf72 families. And again, 2,300 of those visits are made up from individuals with C9. And our main aim in GEMFI has always really been to develop reliable measures that can track people over time and that can ultimately be used in clinical trials. But often one of the problems is that these measures often rely on traditional clinical scales or pen and paper cognitive tests that are administered maybe only annually, that might miss information and lack some sensitivity. So part of the work that we're doing now, as I mentioned, is thinking about how we can use digital technologies and wearables to try and test people more frequently and how these might be more useful for trials. So first of all, when I say digital technologies or wearables, I'm referring to the use of different digital devices. So for example, that might be an app on a smartphone or a tablet, or it could be a smartwatch that you wear on your wrist, for example. And these can either actively or passively collect information about biological functions that would be helpful for our research. So the first benefit we believe, or I certainly believe in using this technology, is that we can gather very detailed and accurate recordings of performance. So for example, we can obtain things like reaction time measures when we're doing these types of cognitive tests that you can't gain from a pen and paper assessment. With those, we can only really look at total scores, for example, which might be missing some important information. So you might be able to get some more sensitive measures from these digital devices. These tools can also be used at home, either on your own devices, or we can provide devices for you, which means that participants in research studies may not need to travel great distances to take part. Again, very convenient for people and also important for those that might be experiencing mobility issues. We may also be able to increase the reach of these assessments to broader populations due to their nature of being able to be completed remotely, and therefore we could have more increased participation, larger samples for our studies and clinical trials. And all of this to say that that is the ultimate aim of this, to help clinical trials. So for these reasons, we developed the EDOF study, which stands for the Early Detection of FTD and ALS, as a digital remote testing sub-study of GEMFI with the aim to further develop and test some of these digital measures to be used in future prevention trials. So there's a lot of information on this slide, so please just bear with me. But there are a whole set of apps, devices and wearables contained within this study that measure the features that we know are important for both FTD and ALS, including motor symptoms. So we've got, for example, a muscle ultrasound and a surface EMG machine that can record activity in different muscles, as well as an accelerometer that attaches to the lower back and can measure different features of gait and walking. We're also looking at language, through speech recordings and extracting different features through automated language analysis. We can look at different thinking patterns, or what we call our cognition, through computerized cognitive apps, but also through other methods like eye tracking. We can analyze different features of behavior, such as empathy, for example, through video analysis of facial expressions using different multimodal AI platforms. And different wearable devices like watches and even now smart rings can measure sleep and activity and also heart rate. And finally, we have some biomarkers of neurodegeneration through portable brain scanning, which I'll tell you more about at the end, and also protein analysis in blood that we hope we can do through blood fingerprint cards, which Alex also mentioned earlier. So a lot of things going on here, and we're at slightly different stages of investigation for many of these tools. So I just thought I'd take you through some of them in a little bit more detail. Starting with a couple of tests of motor function in EDOF. So this is a surface EMG machine study that we're conducting in collaboration with King's College. So we have a protocol that measures muscle fasciculations or muscle activity in terms of frequency and amplitude. And we do that in 3 muscles in the hand, the bicep, and the leg. And we place this sticky pad. I don't have a pointer, but hopefully you can see it on the slide. We place this sticky pad that contains the electrodes onto the skin, and then this little white box records the activity in the muscle. So we want to see if we can see fasciculations in our cohort of individuals that have C9 expansions in particular, and have symptoms of ALS or FTD, or even earlier in those that are asymptomatic. And those individuals are shown here in yellow on the graph. But essentially, this data is showing that in a group of more than 150 asymptomatic participants, we see increased fasciculation frequency in that group, and also in the C9 ALS individuals as well, as you might expect. And these recordings were taken from the muscle in the hand. And important to say that the fasciculations weren't present on visual examination during a standard neurological exam. So potentially, and I say potentially because this is very early data, potentially detecting some early motor nerve changes in these asymptomatic C9 individuals. We also have a small body-worn accelerometer, or IMU, that attaches to the lower back. And now I know what you're thinking, that can't be very comfortable. When I say it is small, it is really very small. It's barely noticeable. I can see someone shaking their head and disagreeing. Perhaps it's not that unnoticeable. But it attaches to the lower back and measures various different features of walking, such as step speed, rhythm, posture, and stride length. And research has shown that a decline in gait speed can be shown many years before the onset of dementia. So we want to investigate if we can also see this in FTD and ALS. And unfortunately, we don't have any data at the moment, but that's where we're heading. We also have this keyboard tapping test, where you simply need to tap as quickly as you can between 2 different keys on a keyboard for 2 minutes. So it's a very quick test, and it can be completed at home on somebody's laptop. And this can detect motor slowness, which, as we know, occurs in ALS and can sometimes occur in FTD too. So early data here, I'm not sure how well that is showing up, but essentially we're showing that we can see differences on this task at the group level between controls and our individuals with BVFTD and ALS that carry a C9 expansion, and also in sporadic ALS as well. But we need to gather a bit more data on this because, as you can probably see by the graph, we have some small samples here. So we're in the process of gathering that. Moving on to cognition, or the way we look at different thinking patterns. So we developed Ignite at UCL, which is an app for the iPad. It essentially contains digital versions of pen and paper psychology tasks that you might complete as part of a clinic appointment or in a research setting. The plus side of Ignite, however, is that it's much quicker, so we can actually get a full picture of a person's cognitive performance in about 20 to 30 minutes, whereas some pen and paper tests can take well over an hour, and I know they certainly do in GEMFI, for example. And Ignite is self-administered. You can do this by yourself at home. As Emma mentioned, sometimes the person that is testing you can really influence how you perform or how you perceive yourself performing. So this is something that you can do by yourself. And what I believe is great about Ignite is that it's also available in 24 different languages. So here on the map, the green countries are where this app is available on the App Store. So 24 different languages in over 60 countries, with the aim of testing more people internationally, which will be important when we hopefully move towards sort of global prevention trials. And Just to show you some data from Ignite, so this is in 4 different tests, or I should say compounded cognitive domains, assessing different functions. So we have performance on the test plotted against disease severity, which is along the bottom of each of the graphs, as measured through a standard clinical scale, where NC, or here on the far left, is healthy controls or non-carriers. 0 represents asymptomatic individuals with no symptoms, 0.5 is mild, 1 is moderate, and 2 or greater is severe. And hopefully what you can see is that as performance on Ignite declines, disease severity increases, which is what you would expect to see. But what we're also seeing is that at the group level, we see statistically significant differences between non-carriers and carriers at different disease stages, even as early as 0. But as Emma quite rightly said, statistically significant differences at a group level. We don't know what this means at an individual level clinically. But promising, nevertheless, that we might be picking up on some of these changes and differences. Eye tracking is another good way of measuring thinking skills or cognition, as we don't need to rely on our hands or upper limb function to test people. So, for example, you don't need to tap and swipe on a screen like you would for Ignite, for example. So it removes that component, which is, of course, important for individuals with ALS. So people simply need to look at images as they appear on the screen, and our eye tracker will gather information. So the eye tracking battery is shown here in the bottom left. So it's fully portable. It's just a very small camera that attaches onto the front of a Microsoft Surface tablet. So the idea being this could potentially be completed at home. And we can output very detailed recordings of eye movements from these devices, which give us insight into somebody's thinking patterns. So here, for example, people are shown different emotions through facial expressions and an emotion word in the middle. People tend to look at the face that matches the emotion. Individuals that have social cognition changes may not be able to do that and recognize that that emotion is corresponding with that word. So this is a measure of what we call social cognition. So this is being completed currently in the GEMFI study, as well as in our symptomatic ALS individuals, but we're working to implement this a little bit wider. Moving on to thinking about how we measure behaviour through wearables. So this is a Fitbit that's worn on the wrist, and it can monitor activity levels. The reason behind using this as a measure of behaviour, or what we call apathy, which can occur often in FTD, where somebody could become socially withdrawn, is that apathy has been shown to be associated with decreased activity levels, as measured by actigraphy watches. So we wanted to see if we could replicate this in our cohort. So for the moment here, we're just looking at simple step count metrics, but we do also have data from heart rate variability, sleep, and everything else that it records, which we're still to analyse. But just looking at this data here, which looks at the peak 30-minute cadence, essentially that is the highest number of steps per minute within a 30-minute time period in both pre-symptomatic and symptomatic FTD compared with controls. And we are seeing statistical group differences there, although perhaps not thoroughly convincing. So we do need to look at that in a bit more detail. But interesting nonetheless for such a simple metric that we can see that. And what I think is quite interesting is that you see a pattern over time when just looking at step count. So this is looking at average step count by day of the week, which you can't see because it's too small, across a 24-hour period. So activity across the 24-hour day is along the bottom of the graph, and the day of the week is Monday to Sunday from top to bottom. And the darker the bar, the more activity or the more steps somebody is doing. But hopefully what you can see is quite clear is the decrease in overall activity levels as you move from being a control to having symptoms of FTD. You see that decrease. And maybe perhaps a particular effect at the weekends. We see that quite prominently, but we haven't looked into this. We don't know why that might be. We definitely need to explore that a bit further. We also are now collecting a lot of this data through our Oura rings, which we're looking to compare metrics with our Fitbit. Some people say they're more comfortable to wear and less annoying. You don't see visual representations of your data through the watch interface, so maybe this is a better option, but that's what we're looking to explore at the moment. And finally, we've just started testing a new portable MR imaging scanner in our participants in GEMFI. So this is called Hyperfine. We want to see if this is a potential remote way of scanning people as part of a clinical trial, for example. So this has recently been acquired at UCL. It operates through the use of an iPad, so you could theoretically scan yourself and just click a button on an iPad and you get an MRI scan. Unfortunately, the quality of the images are not amazing. So on the top line, this is it's a low-field MRI. So this is what comes out, but actually, using AI, you're able to upscale the images to a quality of what you might receive from a standard MRI scan. So that is quite impressive. When I say this is portable, we're not currently sure how portable this is, because although it's on wheels, it does actually weigh a ton. So I can't quite imagine people wheeling it in and out of someone's house, but it's definitely more portable than a standard MRI. We just need to figure out the realms in which we might be able to do that. But anyway, so that was just a brief overview of lots of different things that we're doing at the moment. So we've got several digital tests that assess these features of FTD and ALS. I think it's important to say that I don't think we envisage all of these tools being particularly useful. Perhaps some of them will be useful for FTD, perhaps some of them for ALS. In fact, that's more than likely. We are just doing the work to investigate which of these are the most sensitive and the most helpful, which are the most importantly accepted by participants and the people that use these tools, and hopefully we'll be able to select the ones that are useful for clinical trials. I just want to touch on an exciting project we've just received funding for through the MNDA, where we're looking to expand the use of some of our digital tests in MND by collaborating with other clinics across the UK. So we really want to expand our Ignite and our portable eye tracking in around 100 individuals to establish whether it is in fact feasible and possible to do these types of tests in this cohort of individuals, and if we can detect subtle early change in cognition. And with that, I'd just like to say thank you. Thank you to all my amazing collaborators and funders, but most of all to our participants, caregivers and family members that make this research possible. And thank you to you for listening.